Okay, good afternoon. Today I'm going to present one of our projects of the System Biology group in Senipalma, called Reach-Based Genome-Wide Association Studies and Genomic Selection. Now, well, well, the contents of our presentation are going to be fostering this part. So, our introduction. Well, the first thing that we need to take into account is the nature of the oil palm. The oil palm is a perennial crop. That means that the mean lifetime of the crop is at least 2 years. In our case, this time could be up to 25 years. This makes impossible the implementation of classical breeding programs in this crop. Why? Because the time that we need to get a new generation is too high. In these cases, we use other type of programs, the genomic-based programs. In those programs, we use statistical tools to add the genetic information to the process in order to make faster the obtain of new variants of our plants. But those statistical methods have many problems related with the data. In this case, we're going to deal with high-dimensional data and also with multicollinearity in our data, given by the nature of the genetic information. Our case of study is the height rate. We're going to work with this trait because this is an important trait for the productivity of the crop, because it allows us to extend the productivity lifetime of the crop. To perform those type of techniques, we need two important parts: the genotype of our population and also the phenotype of our population. In general, we are going to use in this study a population of 1,004 vegetal materials taken from the central zone of Colombia in Santander. And we're going to get the genotype of those materials. To get the genotype, we're going to use a classical molecular biology technique as genotype by sequencing. This technique allows us to have a compressed representation of our genome, rescuing the high mutability regions across all the individuals. Using those reads, we're going to use a classical variant calling pipeline to get the differences between our materials. In this case, we implement the GPU accelerated pipeline given by NVIDIA in the toolkit Clara para Bricks that is composed for 4 steps. The first one is the use of BWA to the alignment. The second one, we use GATK for the variant calling. After that, we use GeoNexus to merge that results. And finally, we filter those variants using BCF tools. After that, we have 20,000 variants that display the differences between our materials. For the other part, we have also our phenotype. In this case, we measure the height rate across different years, and we report the mean value for each plant. Yeah. Other thing that is important is that we can create our experimental setup with those information. We're going to separate our data in 3 sets using a classical holdout strategy: train, tune, and testing, given by the 70% of our data set, 15% for the tune part, and the rest for the testing. Okay. About our methods, we divide it in 3 steps. The first one is the complexity reduction. After that, we're going to perform the Genome-Wide Association Studies. And finally, we're going to use those results to perform the genomic selection process. As I said before, one of the most important things in this type of data is that we have high-dimensional data. And also, most part of the future are correlated between them. And that comes from the biological nature of the problem. In this case, we're going to reduce the number of futures using a distance matrix. In fact, a correlation matrix computed using the chi-square test between different SNPs, okay? Using this correlation matrix, we can see that as a distance matrix for a clustering algorithm. In this case, we use an agglomerative clustering algorithm using a cut point of 0.05 corrected by Bonferroni to have the different clusters. We saw that clusters are spatially correlated. And that happens because we can interpret each cluster as a linkage disequilibrium window. And in order to reduce the complexity, we are going to use only one SNP per window. Yeah. In this case, we're going to take the mediate for each window. Finally, after the process, we have 5,000 SNPs that represent most part of the information, approximately. After that, using that data set, we can perform the process of the GWAS and the GS. In the case of the GWAS, I want to talk about the classical approximation and its limitations. In the classical approximation, we use OLS linear regression to perform the adjustment of the model, using also the mean square error as error function to optimize that and to have the values of the effects or the parameters. And with the values of the parameters, we also can compute the variance of those parameters, and taking into account the parameters and its variance, we can compute a classical word test to know the p-value that associates the phenotype and the genotype. The problem of this approximation is that it assumes that the gram matrix of the SNPs is a complete range matrix. But given by the nature of our data, we have this type of data. So we can inverse the matrix, so we can't apply this approximation. One of the solutions that we can take is to perform one regression for each SNP. And it works, but it has some problems. One of the most important problems is that this method tends to underestimate the effects of each marker. So, as a solution, we are going to use a ridge regression to solve that. Using a ridge regression, we can calculate also the covariance matrix, but in this case, we always have a lambda value in the diagonal. That means that the matrix always be invertible. So we can perform the word test across all the SNPs at the same time, correcting that problem. Other thing that is important is that in both cases, we face off the multiple comparison problem, so we need to correct those p-values. In both cases, we're going to use the Story-Tip-Shirani FDR correction method. After the implementation of the GWAS, we're going to implement the GS method, but using also the GWAS. We're going to rank the SNPs using the p-values computed for the GWAS process, and we're going to optimize a ridge regression model using a classical machine learning approach. We are going to use the holdout strategy to optimize the hyperparameters of the model. In this case, we're interested to optimize the number of relevant features associated with the GWAS and the lambda value, the strength of the regularization of our model. Finally, in our results, we see that in the GWAS task, our model allows us to give 71 associated clusters with our trait. And in the GS task, we have 0.63 regression accuracy. Compared with the state-of-the-art methods as from CPU to the GWAS process and RR-blob to the GS process, we notice that we can find more associations, and also we can improve the model performance. So finally, we can say that the model allows us to have a goodness of fit with a polygenic trait as height red. Also, the ridge regression model shows that it could be a promising tool to improve the performance in both tasks, GWAS and GS. Other thing that is too important to take into account is that the ridge regression model is also a biased model. And this type of feature allows us to handle with different problems in our data, like the multiple comparisons. And finally, also the uses of GWAS can be good approximation to improve the performance of GS models. That's all. Thanks for watching.